Welcome back, everyone, to Vox Markets. My name is Paul Hill, and by popular demand, I am delighted again to be able to speak to Alastair Smith, CEO of Avacta Group, a leading biotherapeutics, cancer treatment, medical diagnostics, and COVID testing business. So welcome, Alastair.
Hi, Paul. Thanks for having me back.
Yeah, well, unfortunately, we didn't quite get a chance to cover absolutely everything 3 weeks ago. So hopefully today I was going to run through some of the exciting royalty and licensing opportunities, obviously understanding that there's some commercial sensitivities there and a few other sort of bits and pieces we can see pick up. But before we sort of start digging into the detail, can you just take us through the excellent news last week for the company's lateral flow test that it's developing, that it was able to identify the new COVID strains?
Yeah, of course. Well, I mean, it's straightforward, but obviously very important that the B117, what they call the Kent strain, which is dominant in the UK, and I think now in more than 50 other countries around the world, and the D614G variant, which is the one that very quickly became the global strain. The lateral flow test and the other diagnostics will detect both of those variants. So it takes a little bit of time for us to get hold of new variants when they appear in a harmless form so that we can test them. But now we've shown that the lateral flow test detects those two very important variants as well as the original strain of the virus. And
if there was a sort of like a need to pick up another sort of dangerous variant that changed its spike protein, I presume the Affimer technology could be amended and the reagent to be able to equally recognise and identify those strains.
Yeah, so fortunately the reagents of interest don't bind in the region of the spike protein where the mutations are occurring. But your question is a good one, that there could be a mutation that does affect it. And fortunately the process of generating a new Affimer reagent is really quick. It took us about 4 weeks to generate the first set of Affimer binders to the spike protein. So it's about 4 weeks, but the really important thing is that the lateral flow test architecture, if you like, and its ability to work with nasal samples, that's independent of the spike mutation and the Affimer reagent. So if we need to generate a new Affimer reagent, we can just add that into the existing test strip without having to go back and redevelop the test. And it was relatively easy to get a lateral flow test working in the lab, but it was optimizing it with first saliva and then nasal swabs with no false positives. That took time, but thankfully now we've got a really high quality lateral flow test structure, which we don't need to redo if we need to drop new Affimers into it, or for any future respiratory disease test, beyond COVID.
Infectious diseases, So I mean, basically it's future proofed, isn't it? But I mean, I was looking at the numbers, and then we're talking mind boggling. And I know we mentioned last time that there's 20 million lateral flow tests are going to be required just at schools every week. And I saw there's like 50,000 businesses also going back and have signed up to the government's free lateral flow test. That's probably, I don't know, another 5 million. So we're talking potentially 25 million lateral flow tests over the next couple of months. So, and there was an announcement yesterday from Omega Diagnostics saying that it's sort of like got a contract. It's a sort of like a call-off contract between 50 million and 370 million. So it's absolutely enormous, sort of like, sums that we're talking about. Just can you just quickly remind what the sort of timescales for because I think you mentioned that it was going to be sort of the confirmatory of the on clinical. The clinical trials in about 2 or 4 weeks, something like that, in about sort of a month. Is that sort of still the same timetables?
Yeah, that's right. So we're working through the clinical validation now. We anticipate that we'll have the data to make that CE mark for professional use around the end of this quarter, so a few weeks. The only thing is, getting enough patients now that the numbers have fallen a little bit, but I don't anticipate that being a big problem. So next step is CE marking for professional use. And then to market,
and the manufacturing capacity, I think you said sort of like you were trying to ramp up in the millions a month or something like that, wasn't it? Is that sort of like the indicative sort of the trajectory?
Yeah, so what we've been able to do over the past few months is put in place several partnerships, primarily in the UK, but now looking abroad as well, to be able to ramp that up over a short period of time. So I think that will always be the challenge, supplying such huge numbers of tests. But I think we're in a reasonably good position to deal with that now.
Yeah, good. And again, I'll just highlight to investors over the weekend just how much do these type of businesses go for sort of like, diagnostics. And there was a transaction. Roche bought Genmark Diagnostics on Sunday for £1.8 billion, which I think was over 10 times sales. And they just do an array of sort of infectious disease, and one part of it is COVID. So we're talking big numbers for companies who have unique testing testing technology. So it gives people a sort of like a feel of size. Anyway, sort of like moving to sort of like what we were trying to do, the meat of today, which was sort of like having a look at the third-party royalties and licensing agreements. And I sort of like ran through them, and I mean, frankly, two things sort of came out. First of all, I mean, they just got, an enormous potential sort of like, opportunities out there with a lot of really quite major industry players, which is hugely, sort of like favorable for investors if they want an endorsement, a sort of stamp of approval of the technology. And then I was trying to sort of fit it into sort of relevant buckets. And I sort of like, I came up with a sort of like an idea that, or a schematic of this sort of Olympic rings with the sort of like the two platforms, the Affimer platform and the precision platform in the middle ring of the Olympic ring. And then you've got four major sort of areas. You've got your therapeutics, you've got your diagnostics, you've got your sort of research side of it, and with drug discovery, and then the final bit, which is actually, the purification side. So you've got those four, and they're all sort of overlapping but synergistic in terms of that sort of schematic, and lots of opportunities in between. I was just wondering, other than those four, are there any other sort of rings I'm missing at the top level?
No, I think you've hit on the key ones there. I mean, to be honest, that sort of diagram that you've that you've put forward, we have those sorts of diagrams internally, within the therapeutic piece and the diagnostic piece. But no, you fit on the four sort of main areas there.
Okay, and then sort of like just move on to the specifics on the Affimer platform. Can you just quickly run through top level the key sort of functions that they perform and the key USPs, because it will help people understand why they're so key and the building blocks for a lot of next generation medicine.
Yeah, no, absolutely. So I mean, there's a lot we could talk about, but I'll try and sort of keep it in a nutshell. So the Affimer reagents, then they're proteins that we can engineer to bind to grab hold of a molecule of interest. So to go through your your examples of applications, if you can grab hold of a molecule of interest so you can detect it in some way, then you've got a diagnostic.
Yes.
You can build all sorts of different diagnostic tests,
and that's like the rapid flow, for an example.
Exactly, or the Eliza that we've touched on much, but anywhere where you can grab hold of specifically, so you get a really nice specific test and detect that you've grabbed hold of that molecule, you've got a diagnostic.
Yeah.
And then to take the purification example with Astrea that you mentioned, if you can grab hold of a molecule of interest from a complex mixture, then you can purify that molecule from that mixture, and that's exactly what Astrea BioSeparations want to do. And then
so effectively on that one, you basically, like for the BAMS application, you attach an Affimer to sort of like I think it's a magnetic bead, and then the Affimer, because it's strong and small, then connects with the relevant antigen or the relevant particle and the foreign body you want to do, and then you can separate that Affimer with the foreign body using effectively the magnetic field to then basically purify and leave the existing solution sort of cleaner.
Exactly, and that's one example of how you can use Affimers to purify a product of interest out of a complex mixture. There's other ways of doing it as well, but magnetic beads is a really neat way to do it.
Yeah, and that's important, I guess, for sort of like biosimilar manufacture and other pharma manufacture where you have sort of like natural compounds and you want 100% purification of solutions before you inject them into patients.
Well, that's Astrea. That's their markets, exactly.
Yeah, okay.
Purification in bioprocessing, yeah. The third area I was going to drop onto was, if you've got something that can grab hold of a molecule of interest and block its biology in some way, prevent it from doing what it does, then you've got the opportunity to develop a therapeutic if you can affect the biology of that target that you've grabbed hold of. So that, essentially the Affimer platforms are a replacement for antibodies. A third of all drugs development are antibodies, and the total market, I haven't looked recently, but the total market in life sciences for antibodies must be close to $200 billion.
Yeah, it's absolutely massive.
The key benefits of the Affimer platform compared with antibodies is that Affimers are robust. That they are really robust molecules, so they can be applied to a lot of different scenarios and situations, applications.
And I guess that's really key when it comes to sort of like putting these Affimers in sort of like chemo warheads, where there's effectively a battleground between your immune system and the particular drug, and therefore it's got to be resilient enough to hold up, to allow the drug that you're trying to help the patient with actually improve their condition. I'm guessing has to be actually strong enough to do that.
Yeah, that's an example. The one that I would jump to as an example of robustness is the Astrea example again, because when you make these purification systems, they're not usually a one shot product. You reuse these to purify products multiple times, so you have to clean them, and you have to clean them with quite caustic solutions to clean them properly. So having something that's robust enough to stand up to very low or very high pHs is really important. So there's an example where you have to have something that really is tough as old boots.
Yeah.
Then I was going to say Affimers are very versatile. You can modify them very easily and still produce them at relatively low cost, and they're very specific to the target. So because of the way we generate them, which we won't go into now, we can make them very specific to the molecule of interest and not bind to anything that's quite similar. Then I think the final
no, just on that, Alastair, that's just for investors, that's absolutely crucial, as I understand, for applications for like personalized medicine, whereby you're trying to attach something to an antigen which just unique, so you don't actually attach it to any other cell. So you have to identify a biomarker on the antigen, so they'll allow then the Affimer just to attach to that and no other healthy cell, and then deliver that drug, maybe, I don't know, in a sort of antibody drug conjugate. So it's that absolute, when the company can effectively manufacture an Affimer that is unique just to that biomarker on the antigen, is the critical part of delivering the drug that patient needs to get better.
Yeah, and I would say probably across the antibody markets, specificity is the key issue that needs to be dealt with. Quite often antibodies cross-react because of the way they're naturally evolved to do, cross-react with other targets. You lack specificity. So we very, very commonly with partners and in our own programs come across that specificity issue. So probably that is the key issue with antibodies.
Yeah, and that's absolutely crucial going forward, because for personalized medicine, and now that we're doing a lot more genomic sequencing, and then we've got new sciences like gene editing and CRISPR and all that kind of stuff, having that delivery platform on a unique one-to-one basis is crucial to actually facilitating that whole area of new medical research into just everyday adoption. Otherwise, it won't be able to get that specificity.
Absolutely. The same applies for diagnostics. You want a test that is accurate, if you want to call it that doesn't give you a false positive because of cross-reactivity. You want that in separations because you want to purify your product and not other things in the mixture. So specificity, top of the list. And then the other point I was going to make is that an Affimer is completely unrelated to antibodies. So it's a protein, but it is totally unrelated to an antibody. So quite often we've got freedom to operate around listing antibody IP, which is an interesting commercial angle rather than a technical one.
Yeah, and what about the size? Because it's smaller than your average sort of like antibody. I mean, I've got a bit of form in this area because, unfortunately, I've got a bit of an autoimmune condition. I use, or did use, Humira. It didn't work for me. It's the world's biggest drug. But that's just a straight monoclonal antibody, large molecule, etc. And frankly, and it's a bit like other crude treatments, it can't get to the parts that other, sort of Affimers can. And does a size also help as also an attribute?
Yeah, and we could probably disappear down a bit of a technical rabbit hole here, so I'll resist that. But I mean, just to give you one example of where a small therapeutic can have obvious benefit, and that's in treatment of solid tumours.
Yes.
So you can imagine a smaller therapeutic can penetrate deeper into the tissue, which is an advantage. But yes, small size quite often in a number of applications is also a real benefit.
Yeah. Okay, good. And then sort of like, if we now move into sort of the major sort of royalty areas, the Daewoong Pharmaceuticals, we kick off with that. I mean, just for investors, this is a £1 billion pharma company out in South Korea. And again, I would say that South Korea is sort of like going through a major strategic uplift in its technology on healthcare. So this is another big strategic asset that's getting investment from it. And then, to have a relationship with, sort of Avacta, says a lot about the company's technology. But if you could just quickly at the top level, just take us through the sort of concepts and the sort of like, what we're looking at and how that sort of fits with the sort of like the USPs and, sort of why Avacta was selected.
Yeah. So our relationship with Daewoong Pharmaceuticals is a joint venture called AffyXL.
Yeah.
And AffyXL combines some of Daewoong's technology with the Affimer platform. I'll give you a very quick sort of intro just to put it in context.
Please.
So this is all around autoimmune and inflammatory diseases. I think
you'll get a tick in the box if you can solve this from me.
Well, I wasn't aware of that, but all the more motivation.
Yeah.
I think there's another 50 million people, Paul, worldwide.
Yeah.
With these sorts of issues, and
most of them are unsolved. I mean, Humira is the gold standard from AbbVie, and 50% of people do not get any response. This is and just that 50% is an $80 billion market, enormous.
And you can double that for the total cost of the healthcare that's involved in, autoimmune diseases like rheumatoid arthritis, type 1 diabetes,
Crohn's, colitis.
And then you've got the allergic reactions, asthma, eczema, sinusitis, etc.
And the skin ones.
So the total markets are the total cost of that healthcare is enormous. So let's go to what we can do with Daewoong and AffyXL. So Daewoong have technology around what are called mesenchymal stem cells. So let's call them stem cells for simplicity.
And stem cells are sort of like early doors cells that you largely get in your woman umbilical cord or placenta and stuff. But they, as I understand it, have a unique capability of being utilized to manufacture, or effectively to move into any other different type of cell, because it's so sort of like early days, isn't it, on those cells?
Broadly speaking, that's right. I mean, you can source them from bone marrow and so on as well. And stem cells, mesenchymal stem cells, have some ability to suppress some of these autoimmune inflammatory disease causing immune responses when your immune system overreacts. So they've got real potential, but they are often derived from human material, like we just described, which means you have to continually go back and regenerate them, which is not ideal for getting a supply off the shelf, as it were. And they don't have the ability to counteract all the immune and degenerative signals that that you would like to. So by combining Daewoong's technology, which is a method of getting a renewable supply of mesenchymal stem cells from a single donor. Okay, so that's their technology. They can get a renewable supply, so effectively off-the-shelf mesenchymal stem cells.
Yeah.
What we showed with them, 18 months or so ago, was that you could put the gene, the DNA for an Affimer inside the stem cell, and get the stem cell to actually manufacture the Affimer therapy and spit it out. So that in principle, if you engineer the stem cell to produce, let's say, an Affimer that switches on or off an immune pathway, then put that into the patient, the stem cell will do its thing, but it will also produce the Affimer immunotherapy in situ. You don't have to make the drug, the stem cell does it in situ. So you've got a turbocharged stem cell, if you want to put it in sort of simple terms. Next generation stem cells. That's what we're doing with Affiexcel. And there's
so essentially what you're doing is to say you've basically created an immunosuppressant or immunomoderator. So for somebody like me who's got an over hyperactive immune system that attacks basically anything that I eat, then in theory, with this technology, the stem cell would help effectively the condition by suppressing it. But as I say, that turbocharged using the Affimer on top would help block part of my immune system from attacking that food and causing inflammation and all kinds of different discomfort.
Exactly. And you can, depending on the particular disease area that you're going into, for example, transplant rejection is another one. You would choose what target you wanted the Affimer immunotherapy to bind to. So you can modulate different immune pathways and support the activity of the stem cell. So it's really exciting stuff. It is genuinely next generation stem cell.
I would just again point out to investors on the transplant side, there is just they're so far behind. It's a one fit all. So if you have, for instance, a kidney transplant patient, then the immunosuppressant that they have after the actual operation is just basically trial and luck. But if you have effectively, as you just point out, an Affimer that helps reduce the immune system, but then you also have a test off the back of it, maybe even another Affimer test to instruct the GP or the physician to be able to then adjust the immunosuppressant that they do, then you get, again, the personalised medicine, a much better success rate of that organ, which is in short supply, and you get a longer survival period of that patient who, frankly, because there's just enormous demand for transplant. So, I know it's a difficult one, but when are you going to get the autoimmune sorted out? When's that coming to market? Well, you can't tell, you have to shoot me.
Realistically, these are not rapid developments, obviously, but they are. We hear this word all the time related to Avacta. It's a platform technology. So once you demonstrate that you can engineer stem cells to produce immunotherapies in situ in human with one application, suddenly you've clearly opened up the potential for a whole range of autoimmune and inflammatory disease therapies, cell therapies. So it's you very quickly increase the value of Affiexcel Therapeutics once you've got some human data.
Okay, so then moving on to Astrea BioSeparations then. I mean, given the success, obviously, of the lateral flow, that it's a rapid test. I didn't see on their website that they do things like STD cell. That's the Biokit. Sorry, that it is. Can you just take us through the sort of the separation side, the purification side? Is it?
Yeah, absolutely. So it's. You described magnetic bead separation.
Yes, that's right.
Astrea's market is not magnetic bead separation. What you do is you take non-magnetic beads. So you take beads, you coat the outside of the beads with an Affimer reagent that will bind the molecule of interest, and then you take those beads and you pack them into a column. So that the liquid you flow through has to go over and around all the surfaces of the bead. So you expose all that massive surface area to the molecule of interest, and you flow your complex mixture through. The molecule you're interested in gets captured in the column. The rest of the sample goes away, and then you, as a separate step, can wash out the product that you're interested in. So it's a very standard affinity chromatography process. Astrea focusing primarily in bioseparations, in bioprocessing, as you described earlier. And what we've done with them is that's a multi-billion-dollar market, actually.
Yeah, $2 billion, I think it is.
It's a very significant market. What we've done is we've provided Astrea with initially a non-exclusive license to the Affimer platform for this application. And if they deliver commercially over the next few years, then that can move to an exclusive license with some milestone payments and so forth along the way. So, and the reason that that's in particular a really good deal for Avacta is that we are now tightly focused in the non-therapeutic area on diagnostics. There are, as you described, there are so many different applications where we can use Affimers. You have to focus. Otherwise you end up doing nothing particularly well. So to be able to get a partner like Astrea, a world-class partner like Astrea, to take the platform forward in an application where we are not focused is a really good deal. And then sorry.
And I think again, just a sort of like a broader point for investors, when it comes to sort of anything to do with medicine research and in terms of sort of expertise, then you effectively companies just have to collaborate in terms of, because you cannot just buy expertise, scientific world leaders off the shelf, and therefore you take the best from each company and bring them together. And clearly the Affimer platform is like the building blocks for so many different next generation technologies. Why would it want to try and sort of like compete in blue sky or pay for blue sky research in other areas? Leave that to their experts, and therefore effectively the end result, the end solution leverages the expertise from Avacta with the expertise of your partner, I presume.
Absolutely. It's a mistake to spread yourself too thinly when you've got a platform technology. There's no doubt about that.
Yeah. And then with the Biokit, that was again excellent news last week. And I say that I did notice they do sort of like tests, IVD tests for STDs and rheumatoid arthritis and glandular fever and all that kind of stuff. So again, with an Affimer, I'm guessing with your rapid technology, they could have a turbocharged test, I imagine. That's the sort of theory.
Yeah, and I think the thing I would highlight about Biokit, and those investors who've known the company, known Avacta for a while, our diagnostic business model is two things to it. One is working with partners like Biokit to generate Affimers for them to get into their products, so we can enjoy a royalty on their sales down the line, and the other is to develop our own in-house products. And it has. We've had a large number of evaluations over the last 2 or 3 years, and it's taken a while to get these through to the point of licensing. So, Biokit, Astrea, they're really important validations, really, of the technology, of the business model, the fact that these companies have evaluated the technology extensively over time and are now prepared to enter into a license deal, and then significant cost to them to develop the product and obviously hopefully bring them to market and deliver royalties to us. So I think that for me is the key message, really important validation of the platform. And Biokit in particular is a very well respected, world-renowned diagnostic company. So a great partner, and I think we'll have a broader relationship with them and develop further tests. That's my hope, certainly.
Yeah, well, hey, German engineering, if they can come to it, if they ever come to England for their knowledge and expertise, then it says a lot. And I guess sort of like a bit of a left field sort of like thought and stuff, but sort of with the success of the lateral flow, with this showcasing of the technology, and assuming we get the sort of the right results in sort of like in a month's time or whatever, then it may just even accelerate that sort of whole partnership in the sort of the third party diagnostics area, because people then realize how quick and how effective and accurate the platform is when it comes to sort of like diagnostics.
Yeah, no, absolutely. I mean, there's no doubt whatsoever that the development of a number of different COVID tests, the rapid antigen test being the main one, that's brought a lot of exposure for Avacta and the Affimer platform. It has made the platform very well known globally. And if you just think about what we will deliver shortly, from start to finish in 12 months or slightly under 12 months perhaps, a diagnostic test. That's a hell of an achievement. So yeah, it's brought a lot of attention to the company and shone a spotlight on the Affimer platform, which is great, of course.
Yeah. Well, hey, you set the bar, so for yourselves, you've got to deliver another one after this one as well.
Yeah. I mean, that joking aside, that is obviously the intention. So
yeah, exactly. Yeah.
We're focused very tightly right now on getting the lateral flow test to market. That is our highest priority in the diagnostic business. But we've appointed a product manager who's also thinking about the pipeline of tests that we're developing and bringing those to market. So there is obviously a future beyond COVID, which we are not taking our focus off.
Yeah, and now moving on to sort of like the Affimer Therapeutics. I mean, you've got a marvelous agreement with LG Chem, and I appreciate you can't say much because of the commercial sensitivities. But the two things I would point investors to: just look at the market cap of this business, and it's 46 billion pounds. So we're not talking a small company here. And also, which was really interesting, is I think you've extended the partnership, which says one thing, you've had a major player who likes your technology, and two, they like it so much they've actually upscaled the whole agreement. So can you just take us through again, top level, whatever you can say, non-sensitive in terms of that? Because I think it also talks through the blocking technology that you mentioned earlier on in terms of the unique capabilities of the Affimer.
Yeah, so we are a little bit limited with most of these we can talk about, but LG, their subsidiary LG Chem, which is the pharmaceutical subsidiary, they are a substantial, again, South Korean pharmaceutical company. Our partnership with them is around oncology, autoimmune, inflammatory disease. The potential value to Avacta is quite considerable if all of those programs come to fruition. I think milestone payments, it's the neck end of $400 million. Of course, you've got to be realistic as well, Paul. There's risk in all of these programs, and for all of them to reach clinical market approval would be an astounding achievement. So you have to take some. You have to look at those, bio dollars, as we call them, and just sort of be pragmatic about how you view it. So, but the partnership is really strong. They have, as you said, extended that quite recently. And just a brief word on that, because we can talk about what that is. They've extended that to include our PD-L1 inhibitor. Also to include.
Is a PD-L1 inhibitor basically the Affimer bonding to the antigen, the target that you want to, and therefore effectively blocks the way that it hides from the immune system in terms of sort of cancer. Is that sort of like a layman's explanation?
Yeah, that's broadly correct. So, one of the ways in which cancer cells have evolved to evade the immune system is by sticking the signal on their surface, waving a flag that says, I'm normal, leave me alone. And PD-L1 is that signal. That is what healthy tissue, healthy cells, that's the flag they wave to stop the immune system attacking them, apart from your case, Paul.
They wouldn't survive in my body.
So PD-L1 is that flag. And so if the cancer cell is displaying that flag, the immune system will leave it alone. So if you then create a PD-L1 blockade, so an Affimer that blocks PD-L1, it will stop that signal happening and allow the immune system to attack the cell. So, and then the other area that LG have extended the agreement into is using what we call the Affimer XT platform. And again, we're not going into lots of detail, but it's a really nice platform that we've developed. What it does, it's an Affimer that binds to a protein in your blood, which is very common, called serum albumin. It's a big protein. It doesn't get cleared through the kidneys because it's large. So if you have an Affimer that binds to serum albumin, it will go around in your circulation for much longer. Okay. So then you can attach what you like to that Affimer.
Yeah,
so
okay.
that payload then goes round in your bloodstream for much longer. So it's a way of extending the serum half-life.
Yeah, okay.
A range of therapeutics. So yeah, really nice partnership with LG Chem.
And what sort of like timescales are we talking about extending the half-life? I mean, when I used to inject myself with Humira, I had to do it every 2 weeks. So because effectively the effectiveness went down, and you could tell out in the second week that your condition would be worse than it was in the first. So if you and just for investors, that I used to use Humira, again, a monoclonal antibody. So it's just basically a crude antibody, but that just shows the half-life of an antibody is typically 2 weeks. I don't know, something like that.
That's what you're aiming for. Yeah. No, that's what you're aiming for. So, I mean, you can extend the half-life beyond that, but quite often you don't want to do that.
Yes.
The challenge with many therapeutics is that they have a much shorter half-life, so they're cleared possibly in hours, which obviously isn't what you're aiming for. And so with the Affimer XT platform, it will extend the time that the drug spends in your bloodstream to similar to antibodies, a couple of weeks. Yeah.
And I guess, I mean, the longer it's actually in you, I mean, there might be reasons why you don't want it in there for toxicity purposes, etc. But assuming that that's not the case, then presumably there's lots of drugs that have to be injected or taken once a day or twice a day. And if you can extend the period that they're effective for, it reduces healthcare costs because you don't need a nurse to inject somebody. You don't need people going to hospital and all this sort of stuff. And it becomes a sort of like a, once a month or once every 2 weeks or once every 6 weeks. And that's a really important facet or USP for big pharma or big biotech who want to effectively have their next generation technology.
Yeah, absolutely spot on. No, it's a really sharp question. The concept of taking could be a small molecule drug, could be a peptide drug that has a relatively short serum half-life, so you have to inject daily. And taking that where you can convert that to maybe a weekly or a fortnightly dose, that's a huge benefit to the patient for all sorts of reasons. Yeah,
even just for your skin, because you haven't got to flip and put it into your body every single day. Now moving on to Point Biopharma. Again, it's quite difficult, but I think one of the is it radio pharmaceuticals, etc. But this is again a $15 billion market. I mean, it's colossal.
Yeah. So this sort of really emphasizes why you cannot try to address all these things internally within a company like Avacta. You have to partner and provide your platform to others so that they're frankly using their balance sheets and their resource to do it.
But also the expertise, because I think the critical path is just who's got this scientific knowledge, because it's in short supply worldwide massively, and you can't just buy it in with dollars. It's impossible.
No, certainly not easily. It would be a crazy approach for us to take. So the Point Biopharma partnership is around radioactive therapies, radiopharmaceuticals. So for obvious for oncology purposes, and that is and that focuses around the precision platform, not the actual platform.
Yes, prodrug.
That allows. Again, I've got to be careful what I say, but that allows Point to develop drugs in a similar way that we're developing chemotherapies in prodrug form, so that they are activated in the tumor, to do the same thing with radiopharmaceuticals, so they're released in the tumor and can get into the cancer cells predominantly in the tumor and not damage healthy tissue quite so much. So that's a precision deal with Point.
So effectively, it's almost like lifting and shifting the model you've got for your prodeoxyribosin into radio pharmaceuticals, because essentially it's that sort of the precise way of delivering the drug that you want to the tumor, but whilst it's in the bloodstream and in that transition period, it's just inert and therefore doesn't release its warhead onto the tumor until it hits it.
Yeah, exactly.
Yeah. And then with so moving now to ADC Therapeutics. Now, again, at the point to investors, this is leading edge antibody drug conjugates. And then, I mean, I know a bit about this because I've invested in Gilead, and they spent $21 billion late last year on a company called, I think it was Immunomedics. So this is really, really leading edge stuff. And I think they're only one drug currently, sort of like having been approved, but there are a lot of new partners. Can you just quickly take us through how the Affimer fits in with this? I mean, it's a breakthrough area of medicine.
Yeah, so typically an antibody drug conjugate, which is where the market's been to date, you connect the antibody to the chemotherapy, the warhead, as you called it, with a linker.
Yeah.
And you use the antibody to target cancer cells, so some biomarker on cancer cells. But I think I'm correct in saying all antibody drug conjugates, when they bind to the cancer cell, they have to get sucked into the cancer cell in order that enzymes in the cancer cell will cut the linker, release the chemotherapy, and that will then go and kill the cancer cell. So that's a fundamental aspect of all, I believe, antibody drug conjugates. Now, what we're doing with ADC Therapeutics is replacing the antibody with an Affimer. And I mean, you touched on it earlier on. The key benefit there is small size, deeper tissue penetration, but also specificity. So quite often you might struggle to get an antibody that's specific to a particular target of interest. So that ADC Therapeutics have a set of warheads that they license from AstraZeneca called PBD warheads. Very potent. And so the specific targeting to the tumor cells is really important, so you don't get any off-target effects. But yeah, you're right. Antibody drug conjugates and bispecifics are probably the two key areas in cancer development at the moment.
Yeah, and then with Affimers, when they can get to tumours that other antibodies can't reach. I know it sounds a bit Heineken advert there, but effectively that's it's a unique USP, isn't it?
Yeah.
Now, just
small size, specificity. Those are good. And can I just, Paul, just to
please,
again, for people who know the company, know Avacta well, what I've just described, an antibody drug conjugate, that has to hit a target that is internalized into the cancer cell. Okay, so there's a limited number of targets you can choose. You can't choose checkpoint inhibitors like PD-L1 that we just discussed, because those don't get internalized rapidly. Okay, so that's quite distinct from Avacta's own invention with Tufts University Medical School, the TMAC, which is a drug conjugate, same concept, Affimer, linker, warhead. But now the linker is cut by FAP, which is an enzyme that's outside the cell. Okay, so it doesn't need to get internalized. So the warhead can be released in the tumor microenvironment, not inside the cell. So that lets you pick PD-L1, for example, as the Affimer to target the tumor. So you actually get a therapeutic benefit from the Affimer, as well as the release of the warhead in the tumor. Now that's not what you get with a normal drug conjugate. The antibody, or the Affimer in ADC Therapeutics case, is sucked into the cell and it's destroyed, but the cancer cell is killed by the chemotherapy. So just to put that in context for people, the TMAC concept is fundamentally different and novel in the world of drug conjugates. Yeah,
good. And then just on that one, I know you touched on it again last time, but you sort of like whetted people's appetite in terms of really, big pharma and big biotech who were sort of like looking at this. I mean, how should investors think of sort of like timescales and interest in terms like that? I don't want to put words in your mouth or anything, but clearly it's a unique concept and certainly something that is getting a lot of interest across the medical world.
Yeah, absolutely. I mean, again, pragmatically, Paul, drug development is not it's not a rapid process in the sense of weeks and months.
Of course.
And developing drug conjugates, you can talk to anyone in the field of drug conjugates that that's another step in complexity as well. But you're right. The idea of having checkpoint therapy that is synergistic with the warhead that you choose. So you choose the warhead so that it creates an inflammatory immune response in the tumor. and that recruits the immune system, and you support it with the PD-L1 inhibition. So that is a very novel concept, and we patented that concept actually. So it can't be done with an antibody instead of an Affimer, because our patent.
Wow. Okay, that's clever.
Well, it's a sort of fairly obvious step. If you'd only patented Affimers, then
I wouldn't have thought of that. So yeah, well done.
So we patented the concept.
We
are of course developing Affimer-based TMAC, but we hold the patent around the whole concept.
Antibody.
So it's it'll take us a couple of years before we have human data. But I think there's potential for deal making with preclinical data if it looks really positive in some preclinical animal models.
Yeah. Okay. And then just finally moving on to the Moderna one, the sort of research. That was that final ring we were talking about on the Olympic side. Just going to a point to investors. This is a $54 billion business. It's almost like king of the vaccines because everybody knows it. It does the mRNA technology now. Again, I would point investors, it's all commercially sensitive, so I know you can't talk much about it, but if investors have a look at Moderna's pipeline, it shows how the mRNA technology has been used for mostly vaccines, but also, interestingly, it's been used for immunotherapies, in cancer treatment and stuff like that. And then you could just quickly give us a bit of colour on whatever you can in terms of how the Affimer is being used in the research, because I think you did say, I think it was said in the interims that you were moving into clinical trials. I think not you personally, but I think they were with Affimer. Is that right?
Well, we don't know that.
Okay, sorry.
No, I don't think we said that. So
sorry, it's me.
That's okay. Little bit of background. So Moderna mRNA technology, as you said, can be used for vaccines. Clearly, had a huge impact on their valuation very recently. mRNA can also be used as a way to deliver the gene, the therapeutic protein, such as an Affimer. So you don't need to make the Affimer and inject the Affimer into the patient. You make the mRNA that ultimately encodes the Affimer, you put that into the patient, and the patient's own body makes the Affimer. Okay, so not dissimilar in some ways to what we were talking about with getting the stem cells to make the Affimer. So that is a potentially very interesting, very exciting way of delivering drugs, because mRNA production is a lot simpler, a lot more cost effective than making proteins. So our relationship with Moderna, we generated a number of Affimer molecules against targets they were interested in. We cannot say a great deal about what Moderna are doing. They certainly have Affimer molecules that are of interest, and obviously the objective is that we will support them, hopefully to take something into the clinic through an IND filing. But I haven't got an update I can give you on that right now.
Yeah, I mean, again, I would point to investors. I mean, in theory, my basic understanding of mRNA technology, which they've used in the vaccine area, is it ships it into the body, but it goes into any cells, and then the cells effectively create a spike that then your immune system reacts to, creates antibodies. Now, if you use the same basis with effectively cancer care, and as you say, you genetically or you create your mRNA delivery, if you want it to actually go into the tumor, etc., then you need to attach it somehow to that specificity. So you have to attach it into the tumor, and then if it gets ingested into that particular tumor and then creates a spike, a bit like the operation for the TMAC, i.e. at the surface of the tumor, then it will attract T cells. But equally, if it spits out an Affimer, it can then block on your PD-L1 as well and your checkpoint. So you get a similar sort of theory anyway, that you effectively you stop the tumor from being hidden to your immune system, plus you create essentially another spike that the T cells just say, right, this is really foreign, we don't like this, and therefore come flooding in. But I know if investors want to have a look at the sort of the concepts they're doing, it's well trodden ground on the Moderna the website. Now, just on the sort of the test, the BAMs, Eliza and the Bruker partnerships you have, because obviously there's the healthcare authorities. they're using PCR at the moment, but they haven't got a sort of like a high throughput, really super accurate sort of like lab test in a clinical environment. And I guess that's the sort of like the area that the company is trying to develop a product develop for.
Well, things have moved a long way, as you can imagine, over the last 12 months because of the pandemic. The PCR facilities and throughput has now changed, well, beyond anybody's wildest a year ago. But so with Adeptrix and now with Bruker, we've developed a really good what's called bead-assisted mass spectrometry assay for COVID-19. So this uses a mass spectrometer, which, in fact, I think Bruker probably have the largest installed base of mass spectrometers in hospitals. So the concept is that those mass spectrometers are not being used for COVID testing. And certainly last year, when PCR capacity was under huge pressure, to provide an alternative source of testing capacity, was really promising, and of course Bruker very interested in doing that as well. So we've developed a really good assay with Adeptrix. We're now in the process of getting that into the clinical setting. That assay is with Bruker and with Adeptrix. We've refined the protocols and the methods to suit the clinical workflow, which is important. It has to fit into the process that's used. We're doing that in the UK. Mainly in one particular hospital in the UK, and the machines, the type of mass spectrometers that are in hospitals are considerably, I won't say cut down, that doesn't make them sound very, very good, but they're simplified instruments to allow them to be used relatively straightforwardly in a clinical setting. So we've modified the test to get them onto those types of machines. We're building up a good set of data now on performance, and the performance of the test will ultimately determine how we take that to market. Right now, just to sort of make it clear, our priority is CE marking the lateral flow test.
Yes, good.
As you can imagine, for professional use, and then generating revenue. And then beyond that, we shouldn't forget the potential of a lateral flow test that's CE marked for consumer use, consumer self-testing. Those are different products from a regulatory perspective. So there's a hell of a lot we need to deliver on the lateral flow.
Well, I think focusing on the lateral flow gets a big tick in the box from investors. we might sort of take on it.
The majority of our resources,
and just in terms of sort of the manufacturing capacity for Affimer, etc. Is there sufficient and the sort of the robustness and the repeatability of that whole process? I remember sort of like 10 years ago, they were talking about sort of like how difficult it was to make biologics, and there was the repeatability of it. And I think they've come a long way with biosimilars and stuff, and now those are actually in Europe in terms of big Samsung is making them and this sort of stuff. How easy and how repeatable is it to make sort of like masses and masses of Affimers which will be required going forward?
Well, we touched on that earlier, but that is one of the key benefits of the Affimer platform. It's a really simple protein, actually, and very straightforward to manufacture and purify and get very high yields out of it. So that really is one of the key benefits. We can make Affimers in a very simple system, much simpler than the systems that are required to make antibodies. So, in terms of the manufacturing capacity we have, but both the therapeutic and the diagnostic businesses can produce all of the Affimers they need for R&D, for product development. We've expanded the facilities in Wetherby for the diagnostic business to be able to manufacture Affimer reagents for the COVID testing application. And we can cover the volume required, certainly as far as our own forecasts are concerned, for very, very high numbers of tests. We can cover that in-house because it is relatively straightforward to make Affimers. On the therapeutic side, once you go beyond the preclinical research and you're starting to think about clinical trials, then the Affimer material for that has to be made under what's called GMP So a very highly regulated
Yes.
manufacturing environment, obviously with safety in mind, because you're making a drug product that's going to go into
a human.
A company of our size does not do that in-house. So for the Affimer programs, we outsource that, and we manage that through the clinical development team and the head of CMC, chemistry, manufacturing controls that we have.
Right, okay. So, so for the royalties and the licensing agreements, anything that is basically preclinical, Avacta can manufacture for those programs, whereas anything then goes into clinical human trials, then it goes to a third party who's got all the relevant approvals.
Exactly. And on the diagnostic side, we can manufacture capacity for all of our plans, yeah.
Yeah. Again, just on valuation for investors, just have a look at what medical royalty sort of like, stocks go for. There was a company out in the States, Biospecific Technologies, which went for, I think it was like £500 million just before Christmas. It was bought by Endo International, and that was 12 times sales. So essentially, you've got Roche saying that, basically it's about 1.3 billion pounds, $1.8 billion buying a diagnostic business out in the States. You've also got the royalty streams, which are sort of 10 times sales. And I think you also had, actually, it was quite interesting. I know it was a long time ago, but it is again a bit of a data point. I believe AstraZeneca bought Cambridge Antibody Technologies back in 2006, if I remember rightly, for about £700 million, which in today's terms would be £1 billion. And again, that was on sort of a 10 times sales multiple. So because this is high valuation stuff.
Yeah, I mean, a more recent example as well, Sanofi bought Ablinks for $4.5 billion, I think. So that's
another data point, yeah. And compared to Avacta's market cap with so many different sort of like, Olympic rings, I think the market cap's about £650 million. It just gives you sort of like a sense of scale and opportunity in terms of the huge addressable market. So interesting stuff. Okay, brilliant. Well, thanks very much, Alastair, and we'll definitely, and I am really looking forward to touching base in a couple of weeks' time or 4 weeks' time, whenever the results for the final clinical trials come out for the lateral flow.
Yeah, me too. It's been a pleasure. Thanks, Paul.
Yeah, thanks, Alastair.